Good day, this is Professor Will, CBMD PhD. Today is Friday, March 27, 2020. It is 10:39 and 46 seconds p.m. Eastern Standard Time. And here's a continuation of my lecture on Corona series. So let's get started here. How to identify factors affecting COVID-19 transmission. Date March 26, 2020. Source Stanford University. Summary. Professors describe potential transmission pathways of COVID-19 and their implications. Much remains unknown about the SARS-CoV-2, the virus that caused COVID-19 spreads through the environment. A major reason for this is that the behaviors and traits of viruses are highly variable. Some spread more easily through water, others through air, some are wrapped in layers of fatty molecules that help them avoid their host's immune system, while others are naked. This makes it urgent for environmental engineers and scientists to collaborate on pinpointing viral and environmental characteristics that affect transmission via surfaces, the air, and fecal matter. Researchers are calling for a broader, long-term, and more quantitative approach to understanding viruses such as SARS-CoV-2 that are spread through the environment. There are also principal investigators on a recently announced National Science Foundation-funded project to study the transfer of coronaviruses between skin and other materials, the effect on UV and sunlight on the coronaviruses, and the connection between disease outbreaks and virus concentrations in wastewater. Scientists and medical experts don't have a good understanding of what virus characteristics and environmental factors control virus persistence in the environment. For example, in aerosols and droplets on surfaces, including skin, and in water, including seawater. When a new virus emerges and poses a risk to human health, we don't have a good way of predicting how it will behave in the environment. Part of the problem is historically there has been limited funding for this sort of work. The National Institutes of Health historically hasn't funded work on pathogens in the environment, and funding at the National Science Foundation for this work is limited. Also, coronaviruses and the most emerging viruses that have caught the world's attention over the last decade are envelope viruses that are wrapped in an outer layer of fatty lipid molecules that they've stolen from their hosts. Proteins on the surface of the envelopes that can help these viruses invade the immune system of the organisms they are infecting. There has been much more work on the fate of non-enveloped or naked viruses because most intestinal pathogens in excrement are non-enveloped viruses, like norovirus and rotavirus. Researchers address potential threats that viruses such as SARS-CoV-2 pose to water sources. We usually only worry about viruses in water if they are excreted by humans in their feces and urine. Most envelope viruses aren't excreted in feces or urine, so they aren't usually on our minds when it comes to our water sources. There is increasing evidence that the SARS-CoV-2 viruses, or at least their genomes, are excreted in feces. If infected viruses are excreted, then fecal exposure could be a route of transmission. It is unlikely this could be a major transmission route, but a person could potentially be exposed by interacting with water contaminated with untreated fecal matter. Drinking water treatment systems have numerous treatment barriers to remove the most prevalent viruses and the most difficult to remove viruses. Research on viruses similar to the SARS-CoV-2 virus suggests they are susceptible to these treatments. In terms of virus concentration and persistence, this isn't a worst-case scenario. We tend to study viruses very intensely when there is no outbreak, but the results from one virus aren't easy to extrapolate to other viruses that emerge years later. If we took a broader approach to studying many kinds of viruses, we could better understand the characteristics driving their environmental fate. COVID-19, low risk of coronavirus spreading through tears. Date March 25, 2020. Source American Academy of Ophthalmology. Summary. A new study found no virus in tears of COVID-19 infected patients. While others are certain that coronavirus spreads through mucus and droplets expelled by coughing or sneezing, it is unclear if the virus is spread through other bodily fluids, such as tears. It is unlikely that infected patients are shedding virus through their tears, with one important caveat. None of the patients in the study had conjunctivitis, also known as pink eye. However, health officials believe pink eye develops in just 1% to 3% of people with coronavirus. The study's authors conclude their findings, coupled with low evidence of pink eye among infected patients, suggest that the risk of virus transmission through tears is low. Researchers collected tear samples from 17 patients with COVID-19, from the time they showed symptoms until they recovered, about 20 days later. Neither viral culture nor reverse transcription polymerase chain reaction RT-PCR detected the virus in their tears throughout the 2-week course of the disease. Researchers also took samples from the back of the nose and throat during the same time period. While the patient's tears were clear of virus, their noses and throats were teeming with COVID-19. Researchers say their hopes is to help guide more research into preventing virus transmission through more significant routes such as droplets and fecal-oral spread. Despite the reassuring news, it is important for people to understand that guarding your eyes, as well as your hands and mouth, can slow the spread of respiratory viruses like the coronavirus. Here's why. When a sick person coughs or talks, virus particles can spray from their mouth or nose into another person's face. You are most likely to inhale these droplets through your mouth or nose, but they can also enter through your eyes. You can also become infected by touching something that has the virus on it, like a table or doorknob, and then touching your eyes. Good day, this is Professor Will, CBMD PhD on infectious disease and microbiology. Hope you enjoyed listening and have a great day.